These methods describe the full path of the design, assembly, and characterization of polyelectrolyte complex micelles, which are nanoparticles that are formed from the self-assembly of oppositely charged polymers. Some major challenges with polyelectrolyte self-assembly are avoiding kinetic traps and characterizing the nanoparticles. The salt annealing technique that we describe allows for the repeatable assembly of micelles with low dispersity in both size and shape. And we describe characterization methods, including light scattering, small angle X-ray scattering, and electron microscopy. Delivering therapeutic nucleic acids is a long-standing challenge for nanomedicine. These polyelectrolyte complex micelles take advantage of the strong negative charge of the nucleic acid to sequester them in the core of the micelle, where the neutral polymer corona protects them from nucleases and immune response. The assembly method should be applicable to any type of charged polymers. We've tested them with several polyanions and polycations, and the characterization method should be applicable to any self-assembled nanoparticles, including surfactant nanoparticles and other hydrophobically driven systems.
Begin by incubating the oligonucleotide solution at 70 degrees Celsius for 5 minutes. After the incubation, cool it for 15 minutes at room temperature to thermally anneal the nucleic acids. Then add 40 microliters of 20 millimolar charge concentration diblock copolymer. Vortex the solution immediately and incubate it for 5 minutes at room temperature. To perform the salt anneal, add sodium chloride solution to the oligonucleotide solution for a final concentration of 1 molar. And vortex it for 10 seconds at maximum speed.